হ্যালো। সো একটু লেট হয়ে গেল ভিডিওটা আপলোড করতে, সরি ফর দিস। সো আমরা বেশি কথা না বলে ক্লোনিং ভেক্টর থেকেই শুরু করব। Vectors are DNA molecules that carries foreign DNA segment and replicate inside the cell. They are also called carrier and vehicle DNA. Plasmid and bacteriophages are used as cloning vector. This is because plasmid and bacteriophages have the ability to replicate within the bacterial cell independent of chromosomal DNA. Bacteriophages have very high copies number of their genome within the bacterial cell, but in case of plasmids, some may have only 1 or 2 copies per cell, whereas other may have 15 to 100 copies per cell. Cloning vector use the machinery of bacterial cell replicate thereby increase the copy number, make clones of the DNA inserted into them. This help in easy linking to foreign DNA to allow the selection of recombinant bacterial cells that have picked up recombinant plasmid from non-recombinant those who have not. So then আসি আমরা feature of cloning vector. There are 4 special feature are required to facilitate cloning into a vector. Origin of replication, ORI, যেটা ORI বলি, it is the sequences from where replicate start and any pieces of DNA when linked to sequence can be made to replicate within the host cells. This sequence also controls the copy number of the linked DNA. তো এটা ছিল আমাদের নিট 2020-তে। দিস সিকোয়েন্স অলসো কন্ট্রোল দি কপি নাম্বার ORI মনে রাখতে হবে। এটা আমাদের সিলেক্টেবল মার্কার আসে। দেন ইট হেল্পস আইডেন্টিফাইং অ্যান্ড এলিমিনেটিং নন ট্রান্সফরমেন্ট। সো এটা কী করে? সিলেক্টিভ মার্কার আইডেন্টিফাই আর এলিমিনেটিং নন ট্রান্সফরমেন্ট অ্যান্ড সিলেক্টিভলি পারমিটিং দি গ্রোথ অফ দি ট্রান্সফরমেন্টস। এটাও নিট 2019-এ এসেছিলো, খুব ইম্পর্টেন্ট। Transformation is the procedure through which a piece of DNA is introduced into a host bacterium. Normal antibiotics such as ampicillin, chloraminophysal, tetracycline and kanamycin etc. considered useful selectable marker for E. coli. The normal E. coli cells do not carry resistance against this antibiotic. এটাও নিট 2022 and 20-তে এসেছিল। ক্লোনিং recognition site cloning site, these are used to link an alien DNA. The vector needs to have few or single cloning recognition site for the commonly used to restriction enzyme. The presence of more than one recognition site which the vector will generate several fragments and complicate the gene cloning. Need 2022 এটা। সো এটা আমরা এটা আমি একটা এমনভাবে পড়েছিলাম কি পেস্তা হয়, পেস্তা খেতে খেতে পিভিএস স্যার ইকো R1-এর সাথে ক্লাসে গিয়ে সামথিং কিছু একটা আমার মনে পড়ছে না আপাতত। আমি এরকম একটা বানিয়েছিলাম, বাম সালে গিয়ে কিছু একটা ক্ল্যাপ করছিল, এরকম একটা বানিয়েছিলাম। এটা তো খুব মনে থাকে এইভাবে। তো যাই হোক, এটা হচ্ছে আমাদের ইকোলাই ক্লোনিং ভেক্টর PBR 322। রেস্ট্রিকশন সাইটগুলো দেখাচ্ছে। হেন্ট 3 আর ইকো R1, বাম H1 সাল 1, PVU2, PST1, CLA1 ওয়ারিয়ান অ্যান্টিবায়োটিক রেজিস্ট্যান্স জিন, অ্যাম্পিয়ার অ্যান্ড টেট্রা ROP কোডস প্রোটিনস ইনভলভড ইন দি রেপ্লিকেশন অফ দি প্লাজমিড। এটাও কিন্তু খুবই ইম্পর্টেন্ট। এটা 2019-এ এসেছিলো। তো দেন চলে আসি। দি ligation of foreign DNA is carried out a restriction site present in one of the 2 antibiotic resistance genes. It can be done by H1 site by tetracycline resistance gene in vector PBR 322. In this case, recombination plasmid will lose tetracycline resistance due to the insertion of foreign DNA, but still selected of non-recombinant ones by plating transformant of the medium containing tetracycline. The recombinant will grow in amplicillin containing medium but on tetracycline medium, but non-recombinant will grow on the medium containing both the antibiotic. Hence one antibiotic resistance gene helps of the selecting of transformants, while the other antibiotic resistance gene inactive due to the insertion of alien DNA and selection of recombinant DNA. The alternate selectable marker are developed to differentiate recombinant from non-recombinant on the basis of their ability to produce color of the presence of chromogenic substrate. The DNA is inserted within the coding sequence of the enzyme beta-galactose. This results into inactivation of the gene synthesis on this enzyme and referred to as insertional inactivation. The presence of homogenic substrate that gives blue-colored colonies of this. If this plasmid of the bacteria does not have an insert and the colonies have inserted plasmids, such colonies and recombinated and show no coloration. Vector of cloning gene in plants and animals. A pathogen of several digot plant and Agrobacterium tumerophis is able to deliver piece of DNA to DNA transform normal plant cell, tumor cell and direct this tumor cell to produce a chemical required. Similar, retrovirus animals have the ability to transform normal cell cancerous cell. Tumor inducing plasmid Agrobacterium tumeris can be modified into cloning vector, which is no more pathogenic to this plant, but this still use the mechanism to deliver gene interest into a variety of plants. Similarly, retroviruses can be disarmed and used this to deliver desirable genes into animal cells. Once a gene or DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial, plant or animal host. Competent host for transformation with recombinant DNA. DNA are hydrophilic molecule, so it cannot pass through cell membranes. In order to force bacteria to take up plasmid, bacterial cells must be competent. The competency of ability to take up foreign DNA, the cells can be made competent by following method. Chemical method, CaCl2 method. In this, cell treated with a specific concentration of divalent cation such as calcium, which increases the efficiency with which DNA enters the bacterium through the pores in its cell wall. The cell are incubated with the recombinant DNA and ice followed by placing them briefly at 42 degrees C and then putting them back on ice. This treatment that enables that bacteria to take up recombinant DNA is also known as heat shock method. Physical method, the micro injection. This physical method used to introduce foreign DNA into host cell, and this method recombinant DNA is directly injected of the nucleus of an animal cell. Ballistic of gene gun, another method suitable to introduction of DNA into plants, in which the cells bombard of highly velocities microplastids or gold or tungsten coated with DNA. Disarm pathogen vector. It is method vectors are cause infection of disarm and used to infect the cell and transfer recombinant DNA into the host. Process of recombinant DNA technology.